In this PowerPoint, we are going to cover the following learning intentions from page 17 of your student guide. We're going to describe the role of an enzyme and their properties, describe the function of the active site and its role in specificity of action, and describe the terms degradation and synthesis whilst giving examples of enzymes, their substrates and products. Background knowledge from our previous lesson. We found out in our previous lesson that enzymes function as biological catalysts. Biological relating to the fact they're made by living cells, and catalysts relating to the fact that they speed up cellular reactions and are unchanged in the process. Enzymes are an example of a protein which speed up a chemical reaction. For them to be able to do so, they require an active site. As you can see identified on each of the diagrams, the active site has a particular shape. That shape is complementary to the substrate that the enzyme is going to act on. When a substrate has bound with its active site, we talk about it as forming the enzyme-substrate complex. The active site has a very particular shape. We talk about it being specific. So when a protein is being formed that's going to make an enzyme, the folding of it has to be in a particular way that the active site has a very specific shape. So the shape of the active site of an enzyme molecule is what we call complementary to the shape of its specific substrate. So some examples here, just using hydrogen peroxide, if we look at the diagram. Hydrogen peroxide is a substance that we met in a previous lesson. Hydrogen peroxide is the substrate for the enzyme catalase, which is the enzyme represented in blue. Now, if you look at the shape of hydrogen peroxide, that shape is complementary to the active site on the catalase enzyme. So the hydrogen peroxide would be able to fit into that active site nice and neat. If you look at the alternative enzyme that's in green, which is DNA polymerase, just a random other enzyme, you'll notice the shape of the active site on DNA polymerase is not complementary to hydrogen peroxide. So therefore, those two substances wouldn't be able to bind together. So the shape of the active site of an enzyme molecule is complementary, and we need to use that word complementary to its specific substrate. Again, we've got some examples here on the screen in front of you. Now you'll notice enzyme 1 has a particularly shaped active site, and in this instance it's actually two specific substrates that will be able to fit into the active site, and then we'd see a reaction taking place between them. If we were to take the same substrates for enzyme 1 and try and slot them into enzyme 2 or enzyme 3, for example, you'll notice that they're not complementary. So for an enzyme to be able to function, it has to have its specific substrate, and the substrate binds to the active site because of its complementary shape. We often talk about this specificity of the substrate to the enzyme's active site as being lock and key. So if you think about a key that you use to unopen a lock or a door, that key has a very particular shape for that lock. If you try and put an alternative key into that lock, they don't fit together and they won't unlock the door. The substrate and the enzyme function in the same way. The substrate's shape fits that enzyme's active site and only that enzyme's active site. So if we were to introduce a different substrate to an enzyme, no reaction would happen because they're not complementary to each other. There are two types of reaction that enzymes can be involved in. Enzymes can be involved in what we call degradation and synthesis reactions. The diagram in front of you here represents what we call a degradation reaction, where we see a larger chemical compound being broken down or split into smaller parts. So if we start at the left-hand side of the diagram, you'll see the substrate there is made up of two parts, the green and the kind of orangey colour. The substrate is complementary to the shape of the active site. So they join together, known as the enzyme substrate complex. The enzyme is then going to catalyse the reaction, which sees the substrate broken down into its two smaller parts, and the products are released from the enzyme, freeing up that space to allow another substrate to bind. The second of the reactions that enzymes can be involved in are synthesis reactions. Synthesis reactions involves two or more smaller substrates joining together to form one larger product. So again, starting at the left-hand side of the diagram, you'll see there that we have two smaller substrates separate from each other, but when they combine together, they'll fit into the active site. The synthesis reaction sees products being released on the right-hand side, which are larger than the two substrates were to begin with. Here's an example of some of the common enzymes that you would be expected to know at National 5 level. The first three are degradation, followed by our one example of a synthesis enzyme that we look at in National 5. You'll also see in the table we have the substrates identified and the products. So amylase, for example, is responsible for breaking down starch to form simple sugars, a large molecule being broken down to smaller products. Catalase, we've had a look at catalase previously, but it's involved in degradation. Its specific substrate is hydrogen peroxide, and its products are oxygen and water. We also have the enzyme pepsin, which is a degradation enzyme involved in digestion. And pepsin will take large protein molecules and break them down into their component amino acid molecules. Our example of a synthesis reaction enzyme is phosphorylase, which can be found in plants. Phosphorylase acts on the substrate glucose 1-phosphate, and it builds those small molecules up into large molecule starch. That's the end of this teaching and learning PowerPoint, and I would ask you to reflect and evaluate your own learning based on the three learning intentions that have been covered.